I'm never very good at titles, and when asked to give this title a year ago, this is what I came up with, and actually it still fits what I want to share with you today. Does energetic stress activate mechanisms of proteostatic maintenance and increased health span? But right off the bat yesterday, Dr. Rattan really brought home that this is not an appropriate word for what we study. So I've fully embraced this change and making the term, the dynamic process of maintaining protein turnover, part of how I describe what we're doing. So thank you for that. I learned something in the first 15 minutes that I was here. So hopefully I won't stumble on that term. Another nice thing about going later in the meeting is that I don't need to talk a whole lot about some of these concepts about why we think studying aging is important, because between Dr. Rattan and our first speaker today, a lot of that's already been covered. But I do want to spend some time talking a little bit about why we think understanding protein turnover as part of proteodynamics is important, especially for understanding aging. And in order for you to really grasp some of the data that I want to share with you today, I need to spend a little bit of time talking about our experimental approach. I'm going to share with you just a tidbit of some of the findings that we have from a variety of long-lived models. And then what I really want to spend time on, and the take-home point that I want you all to leave with, is why it's important to consider cell proliferation when we start looking at protein turnover in the context of proteodynamics. Then I'll just make some concluding remarks. So our first speaker showed a version of this in his slides, so I'm not going to spend a ton of time on it. But the whole goal of aging research is really not to increase lifespan. It's to increase the number of years that we have, that we enjoy a disease-free life. To find the pointer. There we go. And so the common approach to doing this has been to systematically understand chronic diseases that we know start to accumulate once we kind of cross the age of 60, 65 years old. And like I said, the common approach has been to know as much as we can about cancer, know as much as we can about cardiovascular disease, diabetes. We've done a good job at understanding each individual age-related chronic disease. What we haven't done a great job of is really expanding this health span, or the number of years that we're living free of the disease, or the burden of chronic disease. So the idea of aging research is really to decrease the number of years that we're living with this burden of chronic disease by understanding biological aging and increasing the number of years free of the burden of disease. So for those of you who do your research in the United States, you know that this is not really our concept. This is a trans NIH initiative called the Geroscience Initiative. And one of the first things that the Geroscience Initiative really did was put out kind of a position paper in 2014 under the direction of Brian Kennedy to really name what are some of the important pillars of aging research. Where should we focus our time in order to make the most progress on slowing biological aging and increasing health span? And so the most important thing to recognize about these pillars of aging research is that every single one of them is connected to the others. So studying one in isolation makes absolutely no sense because they're all interrelated. So even though our lab focuses on proteodynamics and how maintaining proteodynamics relates to our ability to adapt to stresses, our research tends to kind of spill over onto many of these other concepts. So a lot of our work focuses on maintaining metabolic homeodynamics. The points that I want to make today about how cell proliferation is really important in our consideration of proteodynamics makes us start to really recognize what are some of the proliferative cells and how do those change in the tissues that we're interested in with aging or with slowed aging interventions. And of course, talking about some of the damage that hopefully we can prevent by promoting proteostasis, proteodynamics, and adaptation to stress. So let's start by just my quick explanation about why we think that protein turnover is an important aspect of proteodynamics, and perhaps a good readout for understanding whether our slowed aging interventions are successful. So Fort Collins, Colorado is bicycle crazy, and that's why I jumped on this review paper by Nowak and colleagues showing that all of the things involved in proteodynamics are interrelated and interconnected. So we kind of study what I think of as the bookends of proteodynamics, protein biogenesis and protein degradation. But in between those, we know equally important are our ability to take nascent proteins, correctly fold them, traffic them to where their function is going to be in the cell. Yesterday, we spent some time listening to some information about the ER unfolded protein response. We also have a growing appreciation for the mitochondrial unfolded protein response. But our focus is really on protein biogenesis and protein degradation. So why do we think this is important? I love graduate students for many reasons, but they're very good at putting cartoons together. So this cartoon is going to help me to tell you why we think that protein turnover is an important part of proteodynamics. So on your left is going to be a cartoon representing rapid or fast protein turnover, and on the right is slow protein turnover. If you think of each of the ovals as a protein, perhaps a cellular membrane protein for the sake of argument. So we know that some of the stresses associated with aging include reactive oxygen species, advanced glycation end products, and they damage proteins as well as other cell constituents. But if we maintain a rapid rate of protein turnover, this should help us to prevent, over the long run, an accumulation of damaged proteins, as compared to slow protein turnover, where we can have an accumulation of damaged proteins over time, presumably leading to dysfunction. So on the left, the fast protein turnover is what we believe leads to proteodynamic maintenance, as opposed to the slower protein turnover, which leads to an impairment of proteodynamics. So in a sense, we think that maintaining protein turnover is a proteodynamic mechanism that could be important for slowed aging and increasing health span. All right, so real quickly, the experimental approach that we employ to really get our hands on proteodynamics from a protein turnover standpoint. I'll show you some data from cultured cell models, and as was inferred a little bit earlier, a lot of what we do is focused on skeletal muscle, but we also study other tissues. So these are cultured myotubes. I'm going to show you mostly data from murine models. I think in my conversations yesterday, I spent most of my time talking about our passion for using companion animals to start translating some of our findings to human health. Happy to talk about that, but I don't have any data to share. And then we also have human subjects trials as well. So a really common and valuable approach that has been used to understand the biology of aging is to kind of pick and choose genes that we think are important for slowing aging, or perhaps genes that are important for progressing aging, and then do a systematic gain of function, loss of function approach to those. One of the readouts being lifespan. If you're not familiar with looking at lifespan curves, they all look something like this, with percent survival on the y-axis, some aspect of time on the x-axis. In this cartoon, the theoretical genetic knockout enjoys an increase in both median and maximal lifespan. And we've learned a lot from this approach, but it's not the approach that Ben and I took in our lab. And the data that I want to share from you kind of came from looking at a variety of mouse models, all of which shared an increase in lifespan as well as an increase in health span. And we ask, what are some of the shared characteristics of these many different approaches to the same outcome? Because there should be some important information in there. And so the way that we start measuring protein turnover is this. It's quite simple. We use a stable isotope approach where we use an enriched source of deuterium, and the drinking water that we use naturally occurring as deuterium. We use an enriched source, which can then be fed to either our experimental animals, it can be put in our cell culture plates, it can be given to our canine or our human participant volunteers quite safely. And when we do that, then deuterium is then incorporated into newly synthesized proteins via incorporation into amino acids, and also into newly synthesized DNA via incorporation into the deoxyribose moiety. So we can measure rates of synthesis of both protein and DNA over extended periods of time. We then usually take our tissues of interest, and I'm going to show you mostly skeletal muscle today, and we separate them into compartments that we're most interested in. So we can then look at rates of protein synthesis of cytosolic proteins. In the case of muscle, we look at contractile proteins, and then what most of my focus today is on rates of mitochondrial protein synthesis. So this is really a true measure of mitochondrial biogenesis. And then via the relationship between the precursor, and that's the enrichment of the body water pool with deuterium, and the product, that's the proteins in the DNA that are synthesized incorporating deuterium, we can then measure rates of synthesis of both protein and DNA simultaneously. If you're interested in more of the ins and outs of why we think this is a good approach to measuring protein turnover for proteodynamics, we've got a pretty recent review paper. So now I want to show you a few findings from a variety of what I would call energetically restricted rodent models of long life. I don't need to spend any time on the calorically restricted model because everybody in here is familiar with the lifespan extension that is observed across species from 30 to 40% of lifelong calorie restriction. For those of you not familiar with the genetic model that leads to dwarfism, we chose the Snell dwarf model, which results from a mutation of the PIT1 locus and an overall decrease in the growth hormone axis as two of our models. And so I've put those in this kind of grid they'll stay in this position the whole time. So here's our nutritional approach that we chose with calorie restriction, a genetic approach that we chose with the Snell dwarf. Almost as well described as CR these days is rapamycin treatment. So we chose that as our pharmacological approach. So rapamycin is an inhibitor of mTOR, leading to energetic stress signaling. Probably the model that you're not familiar with, because there's only 3 or 4 publications on this now, is the crowded litter model. And it's exactly what it says. If you look at a mouse that's taken from a litter that was only 8 pups and compare it to a mouse from a litter that was 12 to 14 pups, the ones that come from the crowded litter have a transient energy restriction pre-weaning. Competing for mother's milk with 11 litter mates versus 7 litter mates leads to a transient energy restriction. After weaning, they're fed ad libitum, and the result is that these mice have a modest but significant increase in median lifespan, as well as some indicators of increased health span. We're supposing that this is probably an epigenetic model, but this has not really been thoroughly studied. The reason we chose it is we wanted a variety of approaches to increased lifespan and health span. Of note, we have done this in a recent publication with Tracy Anthony with methionine restriction, if you want to look that up. I haven't brought those data. I'm just going to show you a tiny bit of data. All of this is published if you want to look at more of the details. So again, I think of these all as energetic restriction, whether it's truly a decrease in energy intake or if it's via activation of energy sensing pathways. Another good thing about coming late in the day is that I don't need to talk about cell signaling pathways. Everybody in here now appreciates, if they didn't before, that AMPK serves as one of the energy sensors in the cells. And one of its targets is the mechanistic target of rapamycin. So with the models we chose, we have putative AMPK activators, which then lead to TOR restriction or inactivation. We have direct mTOR inactivators. Removal of growth hormone also leads to mTOR inactivation. What you'll see missing here is our crowded litter model. And much to our surprise, when we started doing some of these signaling analyses on the crowded litter model, is that TOR is not inhibited in this transient energy restriction. In fact, in some cases, mTOR signaling is increased in this crowded litter model. So we thought this actually made it a more robust approach because we know that not all of our models share mTOR inhibition as a potential mechanism by which lifespan is extended. So I've placed these all in the same order that I showed you when I introduced the models. Every graph is the same. They're a little complicated, so I'm just going to walk through one of them, and all the rest will be identical. So in the green bars, regardless of the graph, is the increased health span intervention, or the slowed aging intervention. The black bars are the controls, the appropriate controls. On the left y-axis is the fraction of new protein over the course of the study, or the protein synthesis rate. And on the x-axis, as I told you, we like to fractionate our tissues, and in this case, this is skeletal muscle. And my focus today is primarily on this mitochondrial fraction, but there's also a contractile fraction and a cytosolic fraction. So we started by looking at the calorically restricted model, and you can see that the data weren't very, they were fairly underwhelming. Not much of a difference in terms of rates of protein synthesis across all fractions in the calorically restricted model. There was a trend towards a greater rate of mitochondrial biogenesis in the calorically restricted, but not statistically significant. When we looked at inhibition of mTOR, we saw what we expected. Since mTOR regulates protein translation, we expected to see a suppression of protein synthesis rates globally. And we saw this in the contractile fraction and in the cytosolic fraction. What emerged here was that mitochondrial protein synthesis rates were maintained despite suppression of protein translation globally. And that was really the first time that we raised our eyebrows and said something important is happening here. Even though there's energetic stress signaling, mitochondrial protein synthesis seems to be a priority. Moving on to the Snell dwarf mouse, which enjoys the biggest increase in lifespan of all of the murine models. We'll see that across the board, including the mitochondrial fraction, there are lower rates of protein synthesis. And then getting to the crowded litter model, across the board, every fraction, including mitochondrial, greater rates of protein synthesis. So we, of course, we're scratching our heads saying, what do we do next? This is difficult to interpret. It looks like there is no shared characteristic. And that's when we started looking at what's on your right y-axis, and that's the rate of DNA synthesis. So the rate of DNA synthesis reflects cell proliferation. And so what emerges here is that regardless of the intervention, there's either a trend toward or a significantly lower rate of DNA synthesis in this whole tissue. Keep in mind, this might drive some of your questions in the end. This is whole skeletal muscle tissue. So this got us thinking about what are the energetic priorities? Where are energetic resources allocated? And if you look at periods during which growth is not restricted, where the piece of pie is big, we know that much of our energetic demands go towards metabolism. And then roughly speaking, the rest are divided between growth or reproduction and, in yellow, maintenance or repair. But if we start making that pie smaller, either by decreasing energy availability or by activating energetic stress signaling, then this allocation of the other half of the energetic resources gets distributed a little differently, and growth becomes less of a priority, and maintenance and repair becomes a bigger priority. So now I want to spend some time talking about why considering the contribution of cell proliferation is important when we start talking about protein turnover and proteodynamics. And that'll make me revisit some of these data from the rodent models in a minute. So if we just look at the cell cycle, we know that at least in G1 phase, and we're not cell cycle experts, we're having to really start thinking about cell proliferation a lot more now, but at least in G1, but probably also in G2, protein synthesis rates increase in order to, as well as DNA, in order to outfit the daughter cells. Another way to think about this is that during growth, we expand the cell population. So cell proliferation requires doubling of the protein and DNA in order to accommodate these proliferating cells. In contrast, if you look at a period of no growth, or if you look at a cell population that isn't proliferative, then protein energy is allocated differently. So what do I mean by that? We can then look at proteins that can be recycled, broken down, and the free amino acids used to resynthesize proteins and replace damaged and degraded proteins, leading to maintenance and repair, or some people might want to call this somatic maintenance, which is a term that we've known for a long time. So this really made us start considering our protein synthesis data in a different context. So if we look at protein synthesis rates relative to DNA synthesis rates, in this growth situation, this ratio is going to be unchanged, right? Because we're increasing both the rate of protein synthesis and the rate of DNA synthesis to accommodate the proliferating cells. If you contrast that with somatic maintenance, where we have no growth, no proliferation, or relatively speaking, less proliferation, then this ratio is going to be greater because we're increasing protein synthesis without increasing proliferation. So these are the same data that I just showed you, but presenting them as a ratio instead. So now on the y-axis is a ratio of the rates of protein synthesis to the rates of DNA synthesis, and a completely different and consistent story emerges. So every one of the slowed aging interventions in green tends to have or has a significantly higher rate of protein synthesis to DNA synthesis, which we then interpret to mean greater allocation of energetic resources towards maintenance of existing proteins, maintenance of proteins in existing structures. I don't want to be too skeletal muscle centric, so I thought I would throw some heart tissue up here for the cardiovascular researchers in here. I would say that it's even a more pronounced slowed aging characteristic to have this increase in the ratio of protein synthesis to DNA synthesis in cardiac tissue. Sometimes this concept is a little bit easier to demonstrate in a cell culture model because it's simpler. And so these are cultured myotubes that we stimulated from myoblasts to proliferate, to fuse into myotubes. And so in this case, I'm using a rapamycin treatment in culture. And we know that mTOR, when it's inhibited, has an impact on cell proliferation, protein synthesis, as well as aspects of protein degradation. And so what I'm showing you here is mitochondrial biogenesis, the rates of mitochondrial protein synthesis, which in culture we found to be inhibited by rapamycin. Remember, in vivo we saw a maintenance of mitochondrial protein synthesis in skeletal muscle despite global suppression of protein synthesis. However, if we look at DNA synthesis in the rapamycin-treated myotubes, we see a big difference, meaning a much slower rate of DNA synthesis with rapamycin treatment. And if we move on to express that as a ratio of the rates of protein synthesis to DNA synthesis, our conclusion is much different than if we'd stopped here with the mitochondrial protein synthetic outcome. We now have a conclusion that rapamycin leads to reallocation of those protein resources to existing structures rather than to newly synthesized cells. So the last set of data that I want to look at, I kind of added it at the last minute because I kept seeing NRF2 as a transcription factor make an appearance on people's slides in here. And we do spend a little bit of time studying NRF2 ourselves. I'm not going to go through the details of signaling since I'm guessing you're all quite aware that NRF2 transcriptionally regulates a whole host of our endogenous antioxidant network as well as xenobiotic metabolism. What you might not spend as much time thinking about is that there's mounting evidence that NRF2 also transcriptionally regulates many aspects of energy metabolism, including some potential crosstalk with mitochondrial biogenesis. And that's really where our interest in NRF2 began. If you're not aware of the NIH's program, it's specifically funded through the NIA. The program is called the Interventions Testing Program. And while I think we all do agree there is no pill that can increase lifespan and health span individually, the ITP provides a nice platform for people to pitch their ideas on interventions that can be delivered through the diet that might show potential for increasing lifespan. And so the ITP program uses 3 sites in the United States to give kind of a robust approach to measuring lifespan. They do it with a heterogeneous mouse model. And so we proposed a NRF2 activator that I can talk more about in between the sessions, which they tested and found at all 3 sites that only in male mice did this NRF2 activator increase only median lifespan, not maximal lifespan, and not in the females. We're always really excited about sex differences because it gives us a place to start understanding the mechanisms. So the NRF2 activator is what we used then in the last set of data I'm going to show you. These are, instead of myotubes, these are myoblasts. So these are still very highly proliferative muscle precursor cells. Again, looking at mitochondrial protein synthesis. In this case, compared to controls, we wanted to have a stress. So this is a low concentration of hydrogen peroxide, which to be perfectly honest, sometimes when we do this, we see a significantly lower rate of mitochondrial protein synthesis. The data I'm showing you here didn't quite reach significance. So it's highly variable. I'm guessing that's part of in vitro hydrogen peroxide exposure, though. We've done this with a few other stresses as well. And then in the other bar, in the green bar, we have co-treatment of hydrogen peroxide and the NRF2 activator that went through the ITP trial. And what you can see here is that co-treatment with the NRF2 activator actually suppressed the rates of mitochondrial protein synthesis. But then if we look at mitochondrial protein breakdown, so here we can actually directly measure breakdown by looking at loss of the deuterium label from the proteins themselves, or from the DNA. So we see that the mitochondrial breakdown rate is also decreased by the NRF2 activation. If we then look at DNA synthesis rates, the same thing emerges, much slower rates of DNA synthesis, meaning slower proliferation, and the DNA breakdown rate is also much slower with NRF2 activation. So then again, if we look at this as a ratio to try to understand where are these resources being allocated, the ratio of protein synthesis to DNA synthesis with NRF2 activation is much greater than with hydrogen peroxide alone, and the rate of mitochondrial protein breakdown relative to DNA breakdown also much greater with NRF2 activation co-treatment. Kind of a fun thing to point out here in this proliferative cell type, this ratio approximates 1, meaning proliferation can almost completely explain protein synthesis in a proliferative cell model. So just a couple concluding bullets. Everything I just said I think points towards our ability to conclude that activation of energetic stress signaling does seem to activate mechanisms of proteodynamic maintenance in these models of slowed aging and increased health span. And we think it's really important to consider cell proliferation when you're looking at protein turnover as a readout for proteodynamics. Things that we're currently addressing that really arose from this appreciation of how much cell proliferation contributes to proteodynamics, and what might be going through your heads as you listen to some of these data are, we've got a lot to sort out in terms of what's happening with cell proliferation. So again, I've shown you mostly skeletal muscle whole tissue. And so when we're looking at DNA synthesis in skeletal muscle whole tissue, only about 50% of the nuclei are myonuclei. The rest are from other supportive cell types that maintain proliferative capacity. So we currently have no idea which cells can account for this decreased rate of DNA synthesis or cell proliferation with these slowed aging models. So we're doing a lot of cell sorting and trying to figure that out right now. Equally important, we don't know if decreased proliferation is reflective of a greater quiescent phenotype, or if, heaven forbid, it's actually more of a senescent phenotype, which we wouldn't hypothesize since these are all tissues from animals that have increased health span and a longer lifespan. And of course the golden question is, can we target proliferative cell types to improve proteodynamics and somatic maintenance as kind of an approach to slowing aging? Always many people to thank, as well as funding agencies. So I'll try to take some questions if I have time. I knew somebody would ask. So all of the ITP NRF2 activator studies, as well as everything else we've published with it, we've done with Joe McCord. And so it's a commercially available Protandim. Does anybody know Protandim? So it's a combination of 5 synergistic phytochemicals: curcumin, withatherin A, bacchosides, EGCG, and a 5th that's escaping me right now. We're actually on to a 2nd generation NRF2 activator that we're working with Pathways Bioscience, another nutraceutical startup company, and it's in ITP testing right now. Excuse me? They're also phytochemicals. One's derived from luteolin is one of them, rosemary extract, and ashwagandha. Oh, I'm so impressed that I could remember those names under the gun. Only in comparison, we did an Affymetrix array comparing the protandem cocktail to a triterpenoid, CDDO. Sure, I can't remember. And we just kind of wanted to see how similar or different is the gene expression profile. They're very different, far more tipping the scales towards cytoprotection with the phytochemical rather than the triterpenoids. Oh, thank you. Absolutely. I promised to repeat questions and then I didn't do it. Sorry, Mary. So that question was, do we measure the redox state of any of the tissues that we're measuring protein turnover in? In the last 2 years, we've acquired an Ouroboros, O2K, to do high resolution respirometry, but also to measure hydrogen peroxide production. And so that's what we're doing right now.